We talked about the nucleosome. Nucleosome is the histones wrapped with chromatin fibers. So they're called, you see the histone proteins here, and those histone proteins wrapped with the chromatin fiber. Chromatin fiber, this is the fiber of the DNA. So actually they together, histones plus chromatin, they're going to make the nucleosome. Let's talk about the cell cycle now. So what's cell cycle? Is the longest phase. This is the longest phase, the cell cycle. Okay, the cell cycle is the life story of the cell. So you also have the story, you also have the life story, right? And the cell cycle is the same from when it's born, when the cell is born, until it divides into two new cells. This is called the cell cycle or life story of the cell. It has five main stages. The first stage is G1. Okay, the second stage is called S. G1 means growth 1 phase. S means this is synthesis phase. G2 means growth 2. Okay, these three together, G1, S, and G2 together, they call the interphase. Okay, then the mitosis. This is a division of the nucleus, and then the cytokinesis. So stage one, first of all, of the interphase, this is interphase is actually, as you can see, is the longest phase. It is 90% of the cell's life is spent here. 90% of the cell's life is spent in the interphase. So G1 phase. So in the G1 phase, when the cell grows bigger, so we are going to divide the cell, right? In order to divide it, before we have to increase the size of the cell. So cell gets bigger and it makes proteins and the organelles. It does a lot of protein synthesis and organelles. Specifically, do you remember the centrioles? Before this video, we talked about the centrioles. They're going to B2, so they're going to increase in number as well. Performs normal functions like a liver cell doing detox work. And the skin cell, for example, I can say a skin cell making enzymes to break down the bacteria on your cells. This is also example to the G1 phase. So what's S phase? S phase is a synthesis phase when the DNA replicates, makes a full copy. Each chromosome becomes two sister chromatids. In the G1 phase, imagine this is the nucleus of the nematode. In the nucleus of the nematode, we have the chromosomes. But those chromosomes in the G1 phase, all of them are one sister chromatid. Those chromosomes have only one sister chromatid. As you can see here, only one sister chromatid. But what's happening in the S phase, in the S phase, this nucleus, okay, the previous nucleus is going to do what? They're going to do the DNA replication. Okay, they're going to the DNA replication. So after the DNA replication, the chromosomes will have another sister chromatids and they will look like this in the S phase. Okay, each chromosome becomes two sister chromatids. Here, each chromosome is one sister chromatid, but in the S phase, each chromosome is two sister chromatids. Before your bone marrow cell divides, to make the new red blood cells, it must copy all its DNA in the S phase. Okay, this is the example for the S phase. So DNA replication. So we talked about the DNA replication before. We talked about it in the transcription and translation when we were learning about the transcription and translation. So we have the strand of the DNA. And out of that strand of the DNA, what we are going to do, we're going to open this strand with the helicase enzyme. And with the help of DNA polymerase, we're going to synthesize the new strands. We're going to synthesize the new strands of the DNA. And this DNA molecule will fully open and it will have two now. This is the one DNA, this is second DNA. As we increase the number of the DNAs, we are increasing the number of the chromatids that we are increasing the chromatid number. So from one chromatid, we are making second exact copy of that chromatid. So the chromosome now will have two chromatids. It means two sister chromatids. So what's happening in the G2 phase? In the G2 phase, so the cell checks for the mistakes in the DNA in the G2. Okay, are there any problem? Does this DNA have replicated? It's going to check this. Okay, G2 phase checks whether the DNA replication has occurred or no. Okay, and in the G2 prepares everything for the division. Okay, in the G2 you can see the doublet, what? Doublet centrosomes. So this is one centrosome with two centrioles. It's going to double now. After the S phase, it will be also 2, okay? It's starting to double in the G1 phase, but it's completing doubling in the G2 phase. In the G2, so you will have completely 2 centrosomes, 4 centrioles, okay, in the cell, okay? So G2 helps to prevent mutations before a liver cell divides. This is the example of the liver cell. A lot of cells, all the cells do checking in the G2 phase, okay? It prevent the mutations of the DNA, because these G2 phase, we have G2 checkpoints, we check for any mutations, okay? If the DNA is broken, the cell fixes it here and stops dividing, okay? It has two options. G2 phase is going to fix the DNA if there is any problem, if the DNA is broken, if deletions, translocations, insertions happened, or the cell is going to stop division, okay? It's not going to divide, because further division can harm us. So what's happening next? So visible cell during the interphase, the nuclear envelope is intact, okay? DNA chromatin is forming, and the nucleolus is visible, so making the ribosomes now. So at the end of the interphase, what you can see, at the end of the interphase, the nuclear envelope will be still there, so it will be intact. DNA is in chromatin form, okay? DNA is now has two chromatids. The chromosome has two chromatids now, and also nucleolus is visible. But the chromosome is not X-shaped yet because it's in the chromatin form. When it starts to condense, you can see the X shape. But here, after the G2, you cannot see the chromatin form. You cannot see the condensed chromosomes, and you cannot see the X-shaped chromosomes. Okay, I here explained only for the explanation purposes. So this was the stage 1, which is interphase. So what's happening in the stage 2? Stage 2 is actually the mitosis. I also call the M phase, okay? M phase. So this is the actual division of the nucleus, okay? Mitosis is division of nucleus. Okay, this is the division of the nucleus. So this is actual, it has 4 steps. The first step is called prophase. The second is called metaphase, then anaphase, and after that we call it telophase phase. Okay, so prophase, metaphase, anaphase, and telophase. Okay. So let's look one by one. So this is the interphase. We talked that the 90% is spent here. So what do you see at the end of the interphase? I talked to you, right? So the nuclear envelope is intact. So you can see the nuclear envelope still, all right, in the interphase. So DNA chromatin, DNA is in chromatin form. You see the DNA, it's like in a chromatin form. It's like a rope, not X-shaped yet at the end of the interphase. And also nucleolus is still there, okay? And so these are the centrosomes. You can see 2 centrosomes now at the end of the interphase, okay? So here's the video, so you can scan and watch this video. And prophase, metaphase, anaphase, telophase are the stages of the mitosis. So this is also the stages of the mitosis. And let's talk about the prophase. So what's happening in the prophase? So this is the actually the prophase is when the chromatin condense into the visible chromosomes. Now you can see the X-shaped chromosomes here. So the strands were like this, like in chromatin fiber. Now they're going to condense. Okay, this is the condensing, and you start to see the X-shaped chromosomes in which phase? In the prophase. You start to see the chromosomes. The chromosomes are only visible, start to be visible in the prophase okay? And in this phase, the first is chromosome is condensing. Secondly, the nuclear envelope is disappearing. This nuclear envelope start to disappear. So we are disappearing the nuclear envelope. And the spindle fibers are forming here. You see the centrosomes. This is the first centrosome. This is another centrosome. It's made of centrioles, each having two centrioles respectively. And they are synthesizing now what? They are synthesizing the fibers, which we call them spindle fibers. Okay, we call them spindle fibers are forming here. Okay, like a hospital preparing for the surgery. The room is cleaned and the tools are ready. So it's like this. This phase looks like the a hospital which is preparing for the surgery. Okay. So what's happening? The centrioles begin to migrate to the poles. Those centrioles are beginning to migrate to the poles. They are not in the poles yet. This is one pole. This is second pole. Okay, this is equator. Okay, this is equator. So those centrioles are moving to the poles. They are not in the poles yet. They're just moving to the poles. They are starting to make the spindle fibers. They are starting to make the spindle fibers, okay? And the nuclear membrane begins to disintegrate. So nuclear membrane is here. What's happening to the nuclear membrane? It's disappearing, it's disintegrating, okay? So, and the nucleoli is disappearing. This is important. The nucleoli, which is inside, which synthesizes the RNA and the ribosomes. So, sorry, RNA and the ribosomes, the nucleoli, they're going to disappear, okay? We talked about the nucleoli in our first lecture. So centrosomes, the centrosome, you know, this is one centrosome, right? It's in the cell, you can see in this picture, the centrosome. This centrosome has two centrioles, and each centriole has 9 triplets. 9 triplets, third, fourth, fifth, sixth, seventh, eighth, and ninth, right? 9 triplets. So how many microtubules inside? So microtubules, this is one microtubule, 2, 3. Okay, let's count 9 multiplied to the 3, so 27 microtubules. 27 microtubules in one centriole, but in one centrosome I have 2 centrioles. So I multiply 27 into 2, so I will have 54 microtubules in one centrosome. Okay, in one centrosome, I have 54 microtubules. How many microtubules overall in the prophase? So we have 2 centrosomes, okay? We have 2 centrosomes. In one centrosome, I have 54 microtubules. In 2 centrosomes, I will have 108 microtubules, okay? So here you can see, here I have 54 microtubules, and here as well 54 microtubules. So overall, I have 108 microtubules totally in prophase, okay? If someone asks you, you have to answer like this. And each is synthesizing the spindle fibers, or also we call mitotic fibers, okay? So this is all about the prophase. You have to know. If you know these phases, if you know those concepts, they are the same in the meiosis as well. When we learn the meiosis, so you can add what's happening in the meiosis phase, also those 5 processes which are happening. Strands of the DNA becoming condensed. Now you can see the X-shaped chromosome. Nucleoli disappear. Nuclear envelope is disappearing. Spindle fibers are beginning to form. Okay, and the centrioles are migrating to the poles. 5 main things which are happening. So there are 2 or 3 additional things which are happening additional in the meiosis. We will talk about it a little bit later. Metaphase. So this is the second phase of the mitosis. In the metaphase, you have to know 3 things, okay? Firstly, chromosomes are lining up in the equatorial plate or metaphase plate. I can call it metaphase plate, or I can call it equatorial plate. Or equatorial plate, okay? Metaphase plate or equatorial plate, the chromosomes are lining up in the center, in the metaphase plate or equatorial plate. So The centrosomes are already in the poles. Do you remember? This is the second pole, this is the first pole. The centrosomes are in the poles now, and the chromosomes are lined up in the center. We call this center metaphase plate or equatorial plate, okay? You see the centrosomes at opposite poles of the cell, chromosomes lined up in the equator or metaphase plate, and spindle fibers run from the centrosomes to the centromeres of the chromosome. Do you remember in the center of the chromosomes we have the centrioles, we have the centromeres, and those spindle fibers are going to attach to the centromeres of those chromosomes. Okay, they're going to attach to the centromeres. In which process? Of course, in the metaphase. Okay, in the metaphase. So you can see here centrosomes are in the poles now, two centrosomes, and they are attaching, those chromosomes are attaching to the centromeres. So anaphase, what's happening in the anaphase? So anaphase, when the sister chromatids are pulled apart to the opposite poles, so those spindle fibers are going to pull, those spindle fibers are pulling the chromatids. Do you remember they were attached to the centromeres? Now they are starting to pull the sister chromatid. We have two sister chromatid in one chromosome. After the S phase, if they pull it apart, when they pull it apart after that, they will have this kind of looking, and you can see here this kind of look. Okay, so that means sister chromatids are separating. Sister chromatids, I explained what are the sister chromatids in the first video of mitosis. Okay, anaphase is the shortest phase of the mitosis. You also have to know about it. So this is anaphase. Sister chromatids are pulling apart. The question arises, so how many chromosomes here now? Before that, we had 4 chromosomes, right? What about in the anaphase? Now we have 1, 2, 3, 4, 5, 6, 7, and 8 chromosomes. We have 8 chromosomes in one cell now. Okay, we have 8 chromosomes in one cell because we haven't divided the cell yet. We are just dividing nucleus for now. In one cell we have 8 chromosomes. So the diploid number is 4. Here we don't have the diploid number, we have the tetraploid number, which is 8 here. In the human body, the tetraploid will be 92. The diploid will be 46 in human body. Okay, so twice as much. This is the anaphase. In anaphase, the chromosome number increases twice because even though they are one chromatid chromosome, that doesn't mean that they are not chromosome. They are actually chromosomes, but they are only one chromatid. Okay, they will look like this again in the S phase when the new cell divides. Telophase. In the telophase, what's happening? So chromosomes are clustering at the opposite sides. You see the chromosomes are at the opposite poles of the cell. So what's happening next? The nuclear membrane is reforming. You see the nuclear membrane here now. Here also you have the nuclear membrane. So you can see the formed nuclear membranes inside each having diploid number of the chromosomes. Now here we have 4 chromosomes with one chromatid chromosomes. Here we also have 4 chromosomes, each containing one chromatid. Okay, and supercoiled chromosomes begin to unravel. Do you remember the chromosomes were coiled in the prophase? Now they're like this, they're going to be unraveled, so they're not coiled now, okay? And return to their pre-cell division. They're going to return to their pre-cell division condition as long thread-like strands. And the nuclear membrane reforming here, okay? So nuclear membrane is reforming. You see the nuclear membranes here. And one important thing, the cytokinesis is happening here, starting here. Cytokinesis is starting at the end of the telophase, okay? Cytokinesis starting. So this is about the telophase. Chromatids arrive at the poles, right? You see the chromatids are at the poles now. you see the centrosomes in each cell, right? Nuclear envelope is reforming and chromosomes relaxing back into the chromatin phase, okay? This is after that the final stage, which we call the cytokinesis. This is the physical splitting of the cell into two daughter cells. Each gets one nucleus and a full set of organelles. Okay, in humans, it's like cutting one house into two apartments, both fully functioning. This is the cytokinesis phase. Okay. In the cytokinesis, we have two parts in the cytokinesis, which is done by cleavage furrow, and the second is about the cytokinesis in the plant cell. Okay, so you have to know the difference um, between the plant cells and animal cells. Okay, cytokinesis, how the cytokinesis is happening in the animal cells, how it's happening in the plant cells. Let's talk about it. So in the animal cell, cytokinesis is called formation of the cleavage furrow, okay? It means division of the cytoplasm. Actually, the cytokinesis means division of the cytoplasm. Mitosis, this is the division of the nucleus. So division of the cytoplasm is cytokinesis. Division of the cytoplasm into two daughter cells after the nucleus has divided during the mitosis, okay? So cytokinase in the animal cells, it's done by cleavage furrow formation. How it's happening? A contractile ring made of actin and myosin forms just beneath the plasma membrane. Okay, you see, this is the plasma membrane. Beneath of the plasma membrane, they are forming what? They are forming the contractile ring. Okay, here we have the actin and myosin. Okay, it pulls the membrane inward. So this portion is pulling the membrane inward, inside. It's pulling inward, forming a cleavage furrow. This is called a cleavage furrow, okay? The cell then pinches into two from outside like a drawstring bag, okay? So imagine tying a balloon in the middle, you get two small bubbles, right? So if you have one big balloon, if you pull it from the center, so at the end you will have two small bubbles at the end. Here, the cell, in the animal cells also like this. So contractile ring is forming. This is the first thing, okay, by actin myosin. Then it's pulling the membrane inward, okay, forming a cleavage furrow. After that, the cell pinches into two from the outside like a drawstring bag. What about the cytokinesis in the plant cell? Okay, let me give you the example to the cytokinesis. For example, you know about the liver regeneration, right? So regeneration means um, and the forming of the new cell, or restoring of the cells numbers. So liver regeneration after the damage involves rapid mitosis plus cytokinesis in hepatocytes. If the actin filaments are disturbed here, for example, by toxins, the cytokinesis is going to fail, leading to enlarged or multinucleated cells. Okay, this is a problem, real problem. But in plant cells, how the cytokinesis is happening? So actin and myosin here, the pulling inward, right? It's pulling inward the plasma membrane or cell membrane because there is no cell wall in the animals. But in the plant cells, we have the cell wall in the plant cells. Plant cells have a rigid cell wall, so they cannot pinch it inside. Instead, the small vesicles from the Golgi apparatus, they carry the cell wall materials. So Golgi apparatus, small vesicles from the Golgi apparatus, they carry the cell wall materials inside in the plant cells. These vesicles fuse at the center to form the cell plate. You see those vesicles are fusing in the center of the plant cells. Okay, the cell plate becomes a new cell wall. Then they're going to form the new cell wall. They're going to arise from the center to two poles, and after that they're going to separate to two cells. Okay, it's like imagine stacking bricks to build a wall between two rooms. It looks like this. Okay, so what about any examples? So we have a drug which is called colchicine. We use it during the gout, padagra. Okay, were originally extracted from the plants. Colchicine is extracted from the plants and studied because they affect the mitosis or cytokinesis. So plant cytokinesis studies help in creating the anti-mitotic cancer drugs. Okay. So we talked about klutch furrow formation, right? We talked about the cytokinesis in the plant cells, about the cell plate formation. This is called the cell plate formation in the plant cells, cell plate formation. This is one part of the cytokinesis in the plant cells. So if you have any questions, you can approach. So plant cells are building a brick wall from the inside, 2 rooms. Animal cells, they tie a water balloon in the middle, and we are making 2 cells. So these are the quizzes, so you can solve those quizzes in your free time. Okay. If you have any questions, you can approach in the comment section. Thanks for attention. That's all about the mitosis. In the next lecture, we're going to talk about the meiosis process.